Hello, my dear students. I am Dr. Bhaikusri Mohanto, Assistant Professor, Department of Geology, USTM, and today I am going to teach you about a very interesting topic of taxonomic collection, preservation, and identification. So, before starting the topic of taxonomic collection, preservation, and identification, I would like to tell you about the definition of taxonomy. So, what is taxonomy? The term taxonomy was developed from two Greek words, that is taxis and nomia. Taxis means arrangement and nomia means distribution or method. So in simple word, we can say that taxonomy as a branch of science that deals primarily with description of an organism, identification of an organism, nomenclature of an organism, and then classification of that particular organism. Suppose we found one particular living being and we did not know about the identity of that organism. So we first describe those organisms, then with the help of that description, means the outer character, means phenotype of that particular organism, we identify them, and after identification we get a name of that particular creature, and finally we classify those organisms depending on their genetic structure. So taxonomy deals with all these things. They describe the organisms of our world, then identify them, then give them a particular name in the form of scientific name, and finally taxonomy classify those organisms depending on their genetic structure. So my students, this is all about the definition of taxonomy. Classification have a very important role in taxonomy because it makes ranks in different organisms. The classification of organisms have been done according to the hierarchical system or taxonomic rank. So what are the taxonomic rank? In taxonomy, different kinds of ranks are there depending on the genetic structure of organisms, that is kingdom, phylum, class, order, family, genus, species. The organism, those are closely related, we rank them in species. The organism, those are distantly related, we rank them in kingdom or phylum, like that. And in hierarchical system, kingdom is the uppermost rank and species is the lowermost rank. Taxonomy have certain significance. It helps to ascertain the number of living beings on the earth. It aims to classify the living organism. It helps to get an idea of the traits present in plants and animals. It gives the idea of the order of physical development. It gives an idea of local fauna or flora and thus help us to distinguish the endemic species. Now we are coming to our main topic, that is taxonomic collection. So what is taxonomic collection? Biological organism means plants or animals, anything, those we collected, then we preserve them with proper methodologies, and after preservation we documented those organisms with the help of labels and notation, and this whole procedure is coming under taxonomic collection. There are many values of taxonomic collection. In our present slide, we have mentioned some important values of collection. That is, number 1, preservation of extinct species. Number 2, preservation of specimen having special historical value. Number 3, preservation of rare specimen. Number 4, to understand those species found in geographically inaccessible area of the world. So, with the help of collection, we can preserve extinct species. Those organisms which are going to be extinct extinct from our world, if we collect them and preserve them in our museum, it will be helpful to show them in our uh, future students or future generations. Again, those animals or plants having some historical value, if we preserve them in our museum, then it will be very helpful for some historical research also. Then we can preserve many rare specimens there which we cannot found all over abundantly all over our world. So it will be helpful for taxonomic research. And finally, we could not go to different continents of the world because it is money consuming and time consuming. So if taxonomists collect those species found in different continents of our world and preserve them in our museum, then it will be very helpful for our future generation to understand those species and to do some particular research on taxonomy by the students. So these are the main values of taxonomic collection. Now we are going to understand different kind of collecting equipments which are used by different taxonomists. Number one is insect net. Insect net have a wire ring, a bag and a handle. Ring is 12 to 15 centimeter in diameter and made of about 3 mm iron or steel wire. The bag is made of muslin cloth, nylon net or other material depending on the collection method. Killing bottle is attached at the end of the net for Lepidoptera, mean the butterflies and small insects. For aquatic insect or other arthropods, dip net and plankton net is used. So these are different kinds of insect nets which have been used by different collectors. First one is sweeping net. It is basically used for butterflies, Lepidopterans. Second one is plankton net. Phytoplankton and zooplankton collection. Plankton nets are used. Third one is dip net. It is used for fish collection or aquatic insect collection. And finally, Malay strap. Malay straps are used for the aquatic insect collection. With all the insect net, there is another kind of net which is used to collect birds, which is also called as mist net, which you can see in the first photograph, and it is used for collection of birds. Then the next photograph you can see light trap, and this light trap is used to collect nocturnal insect. There is a light present inside this trap, and due to the light the nocturnal insects attract towards it, and after that we can collect those insects. The third photograph is sweeping net, which is used to collect butterflies, and the fourth one is beating net. It is used to collect different kinds of insects found in different canopies of the trees. If we beat those trees with some stick, then all those insects fall down into our beating net. After that we can collect those insects. The second collection tool is aspirator. Aspirator is a suction apparatus used for small insects and arachnids. Transparent plastic vial is used and it is open from one end. The open end have rubber stopper to avoid the crushing of small insects. Two glass suction tubes attached to the rubber stopper. Bulb aspirator are another type of aspirator used to collect mites, small insects and spiders. The third number of collection equipment is Burley's funnel. It is an extraction method of small arthropod from organic soil and leaf litters. It have a metal or plastic funnel having a wire mesh on its bottom for holding samples. The end of the funnel having a beaker with 70% alcohol and a few drops of glycerin. An electric bulb is attached in the upper part of the funnel, providing heat, and due to which the insect crawl downwards and finally fall into the beaker containing alcohol. Number 4 collecting tool is flotation method. This method is also used to extract mites, insects, and other arthropods from soil. This method is used for collecting eggs and pupae of insects from such materials. A sample mixture of magnesium phosphate in water in 1:3 ratio prepared, pour over the sample with insects in a beaker. After stirring for some time, the organism in the sample start floating over the surface of the water, and after that we can collect those insects. The fifth number of collecting equipments is trawling and dredging. This method is used to collect deep sea animals which burrow underwater like mollusks, Urochordata, Hemichordata, etc. Sixth number of collecting tools include killing agents and bottles. We have many kind of killing agents and killing bottles we can prepare. The number one kind of killing bottle is cyanide bottle. It is a white-mouthed bottle with cork and potassium cyanide spread at the bottom of the bottle. Over the cyanide, dry plaster of Paris poured to form a 1.5 to 2 cm thickness. Blotting paper spread over it to absorb moisture to avoid direct contact with the killing agent, because cyanide can make the insect hard and brittle and affects their color if left in the bottle for too long. The second type of killing bottle is ethyl acetate killing bottle. Such bottles are used in hemipterans. Killing bottle is made by using a glass bottle having cotton at the base soaked in killing agent, and then a blotting paper is placed over it. Then the insects are collected in this bottle and the bottle is corked tightly. Third number of killing bottle, that is benzene killing bottle. It is used to kill insect randomly collected from roadside area. To prepare this bottle, a layer of cotton placed at the bottom of the bottle, then thin filter papers need to be kept over the cotton, then 3 to 4 drops of benzene added on the top of the filter paper to kill the insect. 3 to 4 drops of benzene have to be added after 15 minutes of the collection of insect for effective result. After collection, we need to preserve those organisms. For preservation, we have 3 different kinds of preservation technique. First one is dry preservation, then wet preservation, and finally biologically low temperature preservation. In the picture, you can see dry preservation and wet preservation mechanism. In dry preservation, we make a stuffing from the whole animal. The outermost layer means the fur and the skins are there in the animal. We remove all the internal organs and fleshes inside those organisms, and then we stuff those organisms with cotton, and then we preserve those organisms in our museum. Such kind of preservation is dry preservation, which you can see in the photograph. The next photograph, you can see wet preservation, where we use some bottles, and inside those bottles we use some certain amounts of chemicals, and inside these chemicals we dipped our collected organism and preserve them for our future references. As I tell you earlier, for dry preservation, we use animal stuffing method. For wet preservation method, bottle preservation by using chemicals like 10% formaldehyde solution or 10% ethanol solution we use. And for biological low temperature preservation, cryopreservation is there, where we preserve those organisms under -4 degrees centigrade. In wet preservation method, formaldehyde preservation is only used to preserve vertebrates and invertebrates except arthropods. To preserve arthropods, ethanol is used. Formaldehyde preservation is useful for short-term preservation, where preservation of organisms with their color in intact form. For long-term preservation, ethanol preservation is recommended, but ethanol or alcohol dissolve most colors of preserved organism immediately. After preservation of organism, labeling is must because specimen without proper labeling is worthless for taxonomic research or from the collection point of view. For proper labeling, we need recording data. We should record all the data and write down in the collected bottle or in the collected specimen. Then geographical location. We must know about the geographical location where those organisms are collected, and we have to label them. Then we have to label the date of collection, then stage of the organism whether they are male or female or in immature form. Then the collection altitude or depth should be noted, and finally the name of the collector must be noted there. For identification, we must use stereo microscope, forceps and needle for small organisms. Those organisms which cannot identify from our bare eye, we should place them inside microscope and use forceps and needle for spreading the organism under microscope, and after that we can identify that organism very easily. For identify larger organism, we should search different literature, means different books or theses or different sources from internet, and we can use Google Lens in this new world age, and we can also consult with different informants, means different professors, different knowledgeable people about the proper identification. So dear students, this is all about our today's topic, that is taxonomic collection, preservation and identification. I have go through a very good book, that is Theory and Practice of Animal Taxonomy and Biodiversity. It is written by V.C. Kapoor. You can also go through this book for further more information. I have consulted with slideshare.com for the image, and that's all for today. Thank you very much.